Okay, now that I have everything set up, hello everyone. I'm Erin Benson, and like we just said, I'm going to be talking about sulfur isotope heterogeneity at East Eagle. So let's just start it right off with this map that we've just seen from our previous talk. The Eagle and East Eagle nickel copper platinum group element deposits are located right here in the Braga Basin of Michigan near Marquette. The area has Archean-aged basement rocks that are overlain by Paleoproterozoic sedimentary rocks, and the intrusions are part of a dike swarm that's associated with the early stages of mid-continental rifting. Uranium-lead dating places the deposit at about 1107 million years, and I won't dwell too much on the overall geology of the region since we've just heard about that in quite a bit greater detail than what I could go into. So let's zoom a little bit in on the geology of East Eagle specifically. So what we have here is actually two closely related deposits. The Eagle deposit here was discovered first, at least the mineralization was. So previous work has focused a lot more heavily on it rather than East Eagle here. So previous studies at Eagle also looked at sulfur isotopes to identify the source of the sulfur, whether it was magmatic from the Archean basement rock or from the Proterozoic country rock. Those studies found that the semi-massive sulfides had sulfur contamination from the Archean basement rock, and the massive and disseminated sulfides were contaminated by Proterozoic sulfur from the country rock. So our research then focused instead on the Eagle East intrusion. Mineralization at Eagle East was discovered quite a bit later, so it was kind of overlooked. Most of what's actually visible at the surface for this intrusion is an unmineralized melotroctolite. But it was eventually found to be a high-grade nickel deposit, suggesting that, like Eagle, there was likely some crustal contamination. But it was uncertain whether that was similar contamination to what was observed at Eagle, whether the Proterozoic or the Archean sulfur was more influential. So here is a cross-section based on core data. This is a figure that's courtesy of London Mining, so thank you very much to them for this. You can see here at East Eagle this really extensive melotroctolite going down quite a ways, which sort of explains why the mineralization here was discovered a lot later. But there is high-grade mineralization down here, and both of these intrusions are entirely within the Proterozoic rock. East Eagle obviously gets a lot closer to this contact with the Archean basement. And you can see noted here the approximate locations for the samples from this study. You have a very large spatial sampling of East Eagle mineralization. And this also, there's depth to it as well. So you can't see it because it's a two-dimensional figure, but it also goes in and out on the, like into the screen. So very extensive spatial sampling of samples. So we have at East Eagle 3 types of mineralization: massive sulfides. Here's an example from Voisey's Bay, but what we have at East Eagle looks pretty similar to this. Disseminated sulfides, similar to this picture from Duluth, and then, I'm sorry, disseminated sulfides, and yeah, and then semi-massive sulfides. Here's an example from Eagle. Obviously, the ones at East Eagle look quite a bit similar to that. We took a general selection of 50 sulfides from various depths and did multiple sulfur isotope analyses for those samples. That initial selection of samples were primarily semi-massive sulfides. That was just based on the regions of interest at Eagle. But we later found that there were more interesting samples in the disseminated sulfides. So we sampled and analyzed an additional 40 disseminated samples from more laterally extensive drill core to get a better spatial coverage. So, and then finally, we microdrilled a small selection of samples to look at sulfur isotope variability at the centimeter scale. And really quickly, just for anyone who's not familiar with sulfur isotopes, S isotopes are really commonly used to evaluate the source of sulfur in magmatic nickel copper platinum group element deposits. In this study specifically, we analyzed delta 34S and calculated cap delta 33S. Delta 34S is used here to monitor contamination from Proterozoic country rock, which in the area of East Eagle, there's a lot of variability for delta 34S. And then cap delta 33S is a measurement of mass independent fractionation, and it's used to evaluate contributions from Archean basement rock because atmospheric conditions during the Archean caused mass independent fractionation. And so just for comparison to what we would expect for pure mantle values for delta 34S are typically about 0 plus or minus 2 per mil, and for cap delta 33S, 0 plus or minus 0.04 per mil. So we would expect mantle values to be pretty close to 0. So here is the scatter plot for our initial sample of 50 samples that we did multiple sulfur isotope analysis on. I'll start by pointing out the y-axis here, which is our cap delta 33S, our monitor of mass independent fractionation / Archean sulfur contamination. And you'll note that here's our plus 0.04, here's our minus 0.04, and most of our samples are clustered really closely within that range. We have one or two that fall slightly outside of that, but really it's not very compelling evidence for involvement of Archean S. So there's a little bit, but not a lot of compelling evidence that it was heavily involved in this intrusion. We also note that the semi-massive and massive sulfide samples are all sort of clustered together in one region when looking at the delta 34S, sort of in the 2 to 4 per mil region. In contrast, the disseminated sulfides really have a wide range in delta 34S. They go all the way out to 16 per mil in this original set of samples, which is why we looked at the disseminated sulfides in greater depth. So this histogram reflects the distribution of our delta 34S data. The massive and semi-massive data we included as well here as a reminder of what those values were. Again, they're very tightly clustered, 2 to 4 per mil about. So those samples are above the 2 per mil quote unquote limit for what we would expect from mantle sulfur. So this is strong evidence that the sulfur from Proterozoic country rocks was involved in triggering sulfide saturation in the deposit. But the country rock typically contains relatively high delta 34S, and the signals for S that we see in the massive and semi-massive sulfides are comparatively pretty muted. Although obviously still pretty strong evidence that Proterozoic sulfur was involved. In contrast, many of the disseminated sulfides are significantly different and have significantly higher delta 34S. And just in general, more variation. We have one sample down here at -5 per mil and all the way up to 19 per mil. And this wide variation occurs throughout the deposit on a relatively small scale. So within one core, we have variations from 5 to 19 per mil, just within 1 or 2 meters, which raises some important questions about what could cause the difference between the disseminated and massive and semi-massive samples, and why the disseminated sulfides samples exhibit such elevated signals, and why they're so variable on a relatively small scale. So we hypothesize that the wide variation in delta 34S that we observe in these samples is related to many distinct magma pulses that are characterized by distinct delta 34S values. So we could be observing the effects of many magma pulses, potentially even more than I have marked here. I've just done a rough circling of some clusters. So each pulse of magma could interact with a limited set of different country rocks, which could lead to massive amounts of variation on a relatively small scale compared to what we expect. The Proterozoic country rock in the Braga Basin has pretty substantial variation in delta 34S. So multiple smaller magma pulses that are interacting with different rock could logically lead to the variation that we're observing in these disseminated sulfides. We can also turn to the textural evidence here. So our petrographic evidence provides some additional evidence for how this deposit may have formed. We observe several different textures in the deposit, but the most interesting one is this quench texture that's shown here. We have these thin bladed plage crystals that are indicative of a rapid cooling event. And indeed, so large amounts of variation that we're observing within a single core are often related to the presence of this quench texture. So the cores that have those large amounts of variation often have this quench texture present. So I also mentioned before the microdrilling and the analysis of multiple spots on a single sample. So 2 of those examples are shown here in the bottom right corner. So what you can see here is variation of just a few centimeters of as much as 1 or more per mil, which for such a small area is a very large amount of variation. So we have this high degree of variation and we have this quench texture that's present. In theory, what we're suggesting here is that a rapid cooling event could sort of lock in the delta 34S values that are present in the sulfides at the time and prevent later modification of those values. So if we combine this with the idea of multiple pulses of isotopically distinct magma, we could end up with the result of the wide variations that we're observing in these disseminated sulfides. Multiple pulses of magma coming in, forming the disseminated sulfides, rapid cooling, locking in the isotopic signals. So while it's not shown here, it's also worth noting that the massive and semi-massive sulfide samples don't really show any indications of this quench texture at all. So it's not present in those samples. So the quench texture is basically confined to these disseminated sulfides, just like the highly variable delta 34S measurements are. And in many of these disseminated sulfide samples, therefore, our S isotope heterogeneity suggests that the cooling rate is exceeding the diffusive S isotope exchange rate. It's cooling faster than the S isotopes are diffusing. So while this idea of magma quenching helps to explain the variability that we see in delta 34S, it doesn't really account for the tight clustering of the massive and semi-massive sulfides. So to explain that, we're going to turn our attention to the idea of conduit exchange. So this process is basically the interaction between pristine magma and accumulated sulfide liquid. So what we have is pristine magma passing over our sulfide liquid and modifying the original delta 34S values of that accumulated sulfide liquid, bringing them closer to mantle values and helping to homogenize them. So the samples with the quench texture cooled rapidly, so they wouldn't be able to undergo this exchange process with the pristine magma. And so they would retain the small scale variations that are caused by interactions with the country rock. But the areas without this quench texture, i.e. the semi-massive and massive sulfide, would be areas where the isotopic ratios were modified by conduit exchange with the later pristine magma and homogenize them. So this can help to explain why we see such different types of signals in our massive and semi-massive sulfide compared to our disseminated sulfide. So to wrap it all up, because I'm running a little low on time here, what we have in this deposit potentially is multiple magma inputs with distinct delta 34S signatures that are interacting to produce the wide variability that we see in delta 34S ratios, particularly in the disseminated sulfides. And the S isotope variability, even at the millimeter and centimeter scales in these samples, is reflecting a lack of equilibration between the sulfide droplets that's indicating that our cooling rate is greater than our diffusive S isotope exchange rate. And the magmas were likely sulfide saturated when they were emplaced. So interaction with country rocks likely occurred during the magma ascent or within staging chambers in the Proterozoic crust. And just for this research, I'd really like to thank Eagle Mine and London Mining for access to the samples and for helping so much with cross sections and that sort of thing, and also the GSA for funding. Thank you.